Hi, I'm Ben Almquist. I work with Dr. Nick Malash in the Material Science Department at Stanford University. And I do research relating to how do you take materials that you have in everyday life, such as metals and ceramics and whatnot, and get them to interact with cells in your body. And our interest in this is figuring out ways to get these to interact in order to help detect and fight diseases such as cancer. And if you think of a cell kind of like this balloon, it works kind of just to keep everything inside and keep other things outside. And what we wanted to be able to do is, let's say we have our material that we're interested in getting from the outside of the balloon to the inside of the balloon. Well, what we wanted to be able to do is just be able to poke it inside the cell without it bursting. So we want to keep it intact and keep it alive. The problem is, currently, if we take something like a material that's sharp, like a pin, and put it in the balloon, it'll pop. And so what we're trying to do is go back to the former case where what we can do is really allow ourselves to insert something into the cell without damaging it and without it bursting. This is the microscope that I use for my research mainly. It's called an atomic force microscope. And right now it's in this big heavy metal case, and what this does is it protects it from any vibrations from talking or from drafts in the air, things like that. If we open it up, what we see is the actual microscope. And what we have is, this is just your standard optical microscope like you might have used in biology class, just a little bit fancier. And then we have something called the atomic force microscope that sits on top of it. And what this does is it measures the surface of whatever you're looking at. And it works kind of like an old record player where it takes a tip and scans it over the surface. But what we can do with this is combine essentially the height information, or how high something looks like, along with what it looks like optically. So this is the computer that controls the microscope. And what we can do from here is control both the optical microscope and the atomic force microscope. So if we pull up the video image from the optical microscope, what we can see is we can actually turn on some light, and we can pick up a calibration grating that we've put in there. And so here's an example of the kind of image that we can take with this microscope, where what we can see is this is a picture of part of a cell, and the green coloring comes from the optical microscope and tells us where parts of the cell are. And then the height, the 3D part of it that you can see, so kind of along here and whatnot, comes from the atomic force microscope, where it scans out how high everything is. And what we can do is we can take and combine the two and get new insights into what material properties and how things look.